Hi everyone, my name is Manuela Ruiz. I'm a second year medical student and I'm going to be doing a presentation on this article called In Vitro Investigation of Calcitonin Associated Effects on the Trophoblastic Cells. So first of all, I'm going to be doing an introduction on what calcitonin is. Calcitonin is a polypeptide hormone that is secreted by the parafollicular cells of the thyroid gland. This molecule is going to be in charge of regulating blood calcium levels. Well, how does it do this? As you can see in the picture, whenever there is an imbalance in the concentration of calcium, this molecule is going to decrease calcium reabsorption in the kidneys, decrease calcium uptake from the intestines, and then also increase calcium deposition in bones, all of which are going to increase the intracellular concentrations of calcium. On the other hand, what are the trophoblastic cells? So to understand what the trophoblastic cells are, you have to keep in mind that they play a very important role during the implantation period. So as the zygote is moving towards the uterus, it's going to suffer a series of mitotic divisions and it's going to create a blastocyst. This blastocyst is going to divide into two layers, and the outer layer of cells are going to be the trophoblastic cells. These trophoblastic cells will in the future originate the embryonic side of the placenta and are going to be responsible for the secretion of immunosuppressive protein. So for a correlation between calcitonin and the trophoblastic cells, we have that whenever there is an increased expression of calcitonin in the epithelial cells of the endometrium, then you're going to have a decrease in the expression of E-caterines. So what are E-caterines? This is a protein that is part of the adherence junctions between cells, which allows cell-to-cell communication. When this molecule is decreased, there's going to be a dissociation of beta-catenin, which is also part of this complex, and this molecule is going to accumulate in the cytoplasm, and due to a signaling pathway, it's going to be translocated to the nucleus, where it's going to act as an activator of transcription factors that are going to induce cell growth and are going to further promote the implantation process. The objective of this experiment was aimed to determine the effects of calcitonin on the expressions of beta-catenin and phosphobeta-catenin in a dose-dependent manner under the influence of progesterone and estrogen hormones by using jar cell line as an in vitro model for the trophoblasts. For the cell culture, they used jar cells, which they obtained from a choriocarcinoma. A choriocarcinoma is when you have a fast replication of all the cells in the placenta, and this is where they obtained the trophoblastic cells from. After this, they kept them in optimal conditions for the experiment, which was a medium with high glucose, 10% of fetal bovine serum, which is a necessary supplement for cell growth. They also treated them with antibiotics like penicillin and streptomycin, and then they kept them in a humidified atmosphere with 5% carbon dioxide at 37 degrees Celsius. Prior to the experiment, 24 hours before the experiment, they changed this percentage of fetal bovine serum to 2% so they could limit the exposure to exogenous estrogens. After the cell culture, they tested the cells for any bacterial contaminations, and this tested for negative. After this, they divided the cells into 6 different groups. The first group was the control group, which they didn't do anything to. The second one was the cells with progesterone and estradiol. The third group was added 10 nanometers of calcitonin to. Then you had the fourth group with 10 nanometers of calcitonin plus the estradiol and the progesterone. The fifth one, you had a higher dose of calcitonin, being 250 nanometers, and then the sixth one and last, they added to 250 nanometers of calcitonin and the progesterone and the estradiol. So the first method they used to see the effects of beta-catenin and phosphobeta-catenin on the trophoblastic cells was immunocytochemistry, which they used to identify an antigen, in this case the proteins, through a specific interaction with an antibody that you previously labeled. And then after this is done, you stain the cell so that you can see how much presence of that protein you have inside the cell. In this case, the primary antibodies they used were the rapid monoclonal anti-beta-catenin and the anti-phosphobeta-catenin. The secondary antibodies were the biotinylated secondary antibody and the horseradish peroxidase. And as a substrate, they used the amino-ethyl carbazole. So in this study, and as you can see in the picture here, you have the antigens and then you bind a primary antibody. Into that primary antibody, they also bound a secondary antibody. These secondary antibody acted as enzymes which converted the substrate, in this case the aminoethyl carbazole, into a product that would irradiate color so that they could assess the presence of this protein under a microscope depending on how much color was irradiated. The next thing they did was the Western blot analysis, which is used to identify specific proteins from a set sample. The first thing you do is you extract your proteins from DNA and you separate them based on their size and charge with electrophoresis. The proteins in this study were the beta-catenin and the phosphobeta-catenin, which they transferred to a filter, in this case the nitrocellulose membrane, which they had previously incubated with the antibodies that I mentioned. So depending on the reaction between the proteins and the antibodies, you will know how much presence of the protein you have. The last thing they did was a transmission electron microscopy, which is used when you want to see in detail specific parts of a cell or processes going on inside the cell. This microscope uses high-energy electrons, and it has an electromagnetic lens that is in charge of focusing all those scattered electrons and converting all of them into a detailed image. In this case, what they wanted to do with this was see the expression of the adherence junctions inside the cells. For the results, they found out that with a dose of 10 nanometers of calcitonin, there was an increased expression of beta-catenin in both the nucleus and the membrane of the cells, while for the other experimental groups, there was a decrease in the expression of this protein. The next photomicrograph shows us the expression of phosphobeta-catenin, in which we can see that groups C and D have a decrease in the expression of phosphobeta-catenin in comparison to the control group, and that there was also a decrease in the expression of phosphobeta-catenin in the nucleus. Next, we have the Western blot analysis done with electrophoresis, where you see that there was an increase in the amount of beta-catenin whenever you used calcitonin or when you had calcitonin with the progesterone and the estradiol, but these changes weren't significantly greater. And the same occurs for phosphobeta-catenin, where in comparison to the control group, the amount of phosphobeta-catenin in the rest of the experimental groups didn't increase or decrease significantly. And then they also measured the beta-actin, which you can see once again that in comparison to the control group, it didn't change as much. Last, we have the results of the transmission electron microscopy, in which we see that when the cells were given a dose of 10 nanometers of calcitonin, there was an increase in the amount of adherence junctions, which we can see in picture C. And then we can also see that in comparison to the control group, whenever you gave the cells progesterone and estradiol, these levels of adherence junctions also appeared to be increasing. Now for the discussion, we're going to analyze whether what was said by the authors was supported in the experiment. Lee et al. Said that calcitonin is secreted by the endometrial epithelial cells during the implantation window under the effect of progesterone, and this downregulates the expression of E-cadherin, which results in disruption of calcium-dependent adherence junctions. This was not supported by the experiment because we saw that there was an increase in the amount of adherence junctions. Second, we have the opinion of Jitzius and McCalman. They said that trophoblasts are epithelial-like cells and express E-cadherin protein, which is high in cytotrophoblasts and decreases as the cells undergo fusion to form syncytium, while cadherin 11 expression is on increase. This was also not supported by the experiment because we saw that E-cadherin did not decrease its expression in the membrane. We have the opinion of Klaus and Birchmeier said that activation of canonical WNT signaling pathway induces the accumulation of beta-catenin, which in turn translocates to nucleus and regulates the expression of growth and invasion-related genes. This was definitely not supported by what we saw in the experiment. So as a conclusion, it can be said that the usual effect that calcitonin has on the epithelial cells of the endometrium changes when there is an increased production of progesterone and estrogens during the implantation period. And second, we can also conclude that the increased expression of these two proteins And second, we can also conclude that the increased expression of beta-catenin in the membrane and the decreased expression of phosphobeta-catenin in the cytoplasm could account for the amount of adherence junctions that we saw, and thus less trophoblastic cell invasion in the endometrium.